In this portion of Chapter 15, what we're going to look at are the key reactions that are involved in metabolism. And so there are 6 types of reactions that are involved in metabolism. They're going to be throughout metabolism, and there are thousands of different reactions that can be categorized into one of these 6 types of reactions. We can organize all of that crazy metabolic scheme into one of these 6 categories. And so we have oxidation reduction, ligation, isomerization, group transfer, hydrolytic, and then carbon bonds being cleaved by means other than hydrolysis and oxidation. So here's just a table of these reactions and the description of the process that's occurring. We'll get more specific or show examples of these in a second, but oxidation reduction reactions, as you already know, are reactions that involve electron transfer. Ligation reactions, they're ligation requiring ATP cleavage. They form covalent bonds. We have isomerization, which we're rearranging atoms. Group transfer, that's just taking a functional group and transferring it from one molecule to another. Hydrolytic, we're cleaving bonds using water. And then those carbon bond cleavages by means other than hydrolysis or oxidation. Here we have 2 substrates that will yield one product, or one product that will yield one substrate rather that will yield 2 products. And so we're going to go through and look at each of these individually. So oxidation reduction reactions rather. These reactions are essential for many of the pathways that we'll look at. So here what we're just looking at is an example of electron transfer. And I do want to point out, note that in this case, in both of these cases, we have that electron carrier that we've talked about present. So remember that in redox reactions, one substance is oxidized and loses its electrons, the other is reduced and gains the electrons. So remember that the FAD and the NAD plus, these are going to take the electrons in addition to hydrogen, but they're gaining the electrons, and the other substance, whether it be succinate or malate here, is going to lose the electrons and transfer it to that electron carrier. And we've already seen why these electron carriers are important. With these ligation reaction, these reactions form bonds using energy that comes from ATP cleavage. So you have your substance and you have ATP, and in the process, this is a coupled reaction. You take this ATP and use water to hydrolyze the ATP or to break off that phosphate. That energy that comes from taking this ATP and breaking it apart is going to be used in order to take this substance and combine with this other one. So we go from pyruvate to oxaloacetate because we can couple this to ATP and use the energy of that ATP cleavage in order to ligate or bring together 2 other substances. So here we have CO2, carbon in the form of CO2, being added to that pyruvate molecule. Isomerization reactions, these reactions are rearranging the atoms that are present in a molecule. Typically, this is going to occur, it's an important step, because it can actually take a molecule and prepare it for a subsequent reaction. And one of the, not more common, but one of the more prominent cases of this isomerization is the conversion of citrate to isocitrate. And so the citrate is in a form that is not ready to undergo a particular process, so we make some rearrangements. There's an isomerization. The atoms are rearranged within the molecule in order to form the isocitrate, which then would go on to the next step in a process. Group transfer reactions, they include a number of different types of transfer substances, but one of the more common would be that phosphoryl transfer process that we've talked about. And so here we're showing ATP and that phosphoryl transfer of ATP. Here that phosphoryl group is going to be transferred to glucose, and we make that glucose 6-phosphate, and ADP results. So we've seen the significance of the phosphoryl transfer and its role in metabolism earlier in the chapter. Hydrolytic reactions, here we are cleaving a bond by adding water. These types of reactions are commonly used to break down larger substances. In this particular example, we are breaking a peptide bond by adding water. So the peptide bond is broken here. You use water to cleave this bond, and you end up with two substances. Then we have carbon bonds that are cleaved by other means. Lyases are enzymes that will catalyze the cleavage of carbon bonds by means other than hydrolysis or oxidation. So lyases. Here, the example that we're looking at shows the breakdown of fructose 1,6-bisphosphate, and it breaks it down into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. We'll come back to this reaction in our study of glycolysis, but it's a lyase that is going to help this process occur. So we're not using oxidation or hydrolysis here to break this substance into two other substances. There's an enzyme, a lyase, that's going to come in and help that process occur. So we've looked at all of these reactions. There are 6 of them, and we've looked at the examples of 6 different reactions. Again, just examples where these would occur. But because we have all of these reactions and they're organized into these categories, we also need to be thinking about how do we regulate the process of metabolism. And there are really 3 principal ways that we can regulate the metabolic process. We've seen some of these before because we've talked about regulation of enzymes. How do we control enzymes? Well, one way is to control the amount of the enzymes that are present. So enzymes are going to be involved in a lot of these processes. So how do we control the amount? Well, we've said in previous chapters that this is done on the level of transcription. We can control the catalytic activity, and that control of catalytic activity, there are a variety of different methods that we can use to achieve this. We can have allosteric control. We can have reversible covalent modification. We can have hormones being involved, and there are other means, but these are just some examples of ways that we control catalytic activity. And then we can control the accessibility of the substrates. The way that this is achieved would really be through compartmentalization. Basically, if we physically separate out opposing reactions, we can better control the reaction system overall. And the idea is to get to a state of homeostasis. And in that case, we have balance between all of these different processes that are occurring. So this is just another way of looking at this metabolic pathway. We talked about bringing food in and how it's broken down into some biomolecules that then undergo more detailed metabolism. So this is just kind of showing balance between some of these different reactions that are taking place here. All right, so that wraps up chapter 15, and this information, again, what we've looked at are the key reaction types, and there are 6 of them. So the 6 types, can you name those 6 types? Can you tell me what's involved in those 6 categories of reactions? Can you tell me an example of how that is used? Also, we looked at how do we regulate these processes that are occurring, and we said that there are 3 main ways to regulate these. We looked at controlling amount, controlling catalytic activity, and controlling accessibility. So for each of these, how do we do that? At what level do we do that? And then the ultimate goal is to maintain balance within these systems.